Okay, thanks everyone for coming along and watching live. And I know a lot of people are going to be watching this online as well. I'd just like to thank Linda for offering to give the talk today. And she'll be talking about some work looking at ancient DNA from Antarctic marine sediments and also some of the cool techniques she's been using to study that. So I think we'll probably get underway if that's okay. Yep. I'll hand it over to you, Linda.
Cool. Thanks, Jamie. Yes. I'll be talking about ancient DNA from Antarctic sediments, primarily, or most of the first half of the talk. And then Mary-Eve has asked me to talk a bit about hybridization capture. So the second part of the talk will be mostly about that technique. I'll just start with Antarctica here, or generally just a quick recap on why we're doing ancient DNA in the marine environment. So obviously it's a really cool technique to look into the reconstruction of past marine communities, because we can look into all sorts of organisms that don't necessarily fossilize. So bacteria, archaea, eukaryotes. My primary interest is in phytoplankton, zooplankton, in that sort of area. But there's some interesting signatures of higher organisms as well, which would be really interesting to look into. But when we're working in the marine environment, and you can already see on the picture here, like we have quite a bit of a water column that we need to work through first to get to the sediments. And so contamination in this field is quite a significant problem that we need to address. And therefore, we try our best to authenticate the sediments that we're pulling out of the deep ocean. And that can be done with fragment analysis or with damage analysis. And I'll talk more about this in a couple of slides. First, study Antarctic environments in particular. So that's my main focus region at the moment. And not just because I like to go there so much, but also because it's a really interesting environment and important, of course, in the context of climate change. So when we look at seafloor sediments in this region by the different types of communities that we can find, we can, for example, make estimates about how far sea ice has extended in the past, or what looks more like an open ocean community. And so we can make some estimates about past the Antarctic environment and the sea ice. Then the very deep ocean is a good environment for DNA preservation. We have very cold temperatures in regions, low oxygen concentrations, no UV radiance, and so these are all factors that contribute to good DNA preservation. Then, of course, that's a bit region specific, but in many areas we have some undisturbed sediments, which gives us quite a nice layering and record of the past. And then what's also important to consider in terms of where we want to core is whether we have high or low sedimentation rates. We're restricted to certain types of coring systems in the deep ocean. And so depending, or say if we had low sedimentation rates and we have less accumulation of sediments over time, then that means with a coring system of a specific distance, we can reach further back in time because we have a shorter distance that we can look at. So these are all factors we consider and that make the Antarctic environment quite interesting to study. Most of the first couple of slides that I'll be showing in data is from IODP Expedition 382. So this is an expedition I was a participant on that happened in 2019. You can see me here Can you see my mouse? Not sure, but I'm the one with the little white beanie there to the side. So this was a 2-month expedition to the Scotia Sea in 2019, and the longer title of this was Iceberg Alley, Subantarctic Ice and Ocean Dynamics. Many of you might not be familiar with the International Ocean Discovery Program, and so I thought I'll just briefly show you what that's about. So the IODP is basically, it's the largest geoscience program that exists. It runs since approximately 50 years. And on the map here on the left, you can see all sorts of coring locations that have been locations that have been cored in the past. So there's a lot in the marine environment. And basically every expedition gets a running number once it gets scheduled. And so our expedition was number 382 in that sequence. We left from Punta Arenas and then traveled through the Drake Passage here towards the tip of the Antarctic Peninsula, and this region is the Scotia Sea. And so you can see a couple of tiny labels here on this map. That is because before we start heading there, we already have a good overview about which sites we're attempting to core. So we're not blindly going somewhere and taking a core. There's a lot of pre-existing information about the sites, like what's the bathymetry like, the seismic information, so that we have a good idea of what the seafloor looks like in the areas where we're actually targeting to put a sediment core down. So in the end, we were coring 5 sites, and 3 of those I've labeled in bigger here, because those are the 3 sites that I'll be showing you some data from. So there's the Central Scotia Sea, the Southern Scotia Sea, and a bit of the data from the Falkland Plateau as well. In case you're interested in what the IODP is doing, you can look that up. The ANZIC is the Australian New Zealand branch of the IODP, and they have a website, and there's lots of opportunities to get involved, whether that's on expeditions or training courses for students, or all sorts of other opportunities. It's always advertised there. So yeah, just thought I'll let you know about that. And then one of our outreach officers on board, Marlo Garnsworthy, she's actually put together this really nice animation of the region and why it shows very nicely why we would want to core in an area or in this Iceberg Alley area. So just look at the time here. And so this animation shows you how icebergs are being pushed off into the ocean, and how they're traveling around the Antarctic continent, and they go into this region here that is called Iceberg Alley. So I just let that run. So what you're seeing is that with time, more and more icebergs in recent years were being pushed into the ocean due to warming. And so Iceberg Alley has become a really active area in terms of iceberg travel. And so it's a really great location to put a coring system down, collect sediment cores, and look at the evolution of the Antarctic ice sheet over the past. So that's why we went there. And then the ship that we were using to do all this work is the JOIDIS Resolution, was the JOIDIS Resolution. So this big drill ship here is basically a floating drill rig with lots of labs, a little bit of accommodation, and some food. And so we're working in the deep ocean, and you can see again here, this is the equipment that we need to put down. So what you don't see on this picture here is there's a huge hole in the ship. It's called the moon pool, and it's under the drill rig so that the coring equipment can get lowered down through the ship into the ocean, and we're pulling these sediment cores out of the seafloor. That's pretty much a real life image of what it then looks on deck near the drill rig. So we have these really big coring pipes, 30 meters at a time, that get sort of screwed together and put down through the water column bit by bit. And we were operating in about 3,000 meters of water. So you can imagine all of this is quite some extensive logistics, and it takes a lot of time to put all of this together. And so that is why these expeditions take so long, 2 months at sea for coring 5 locations, really. Just a few more images of the ship. Here you can see the drill rig again, and once the sediment cores, they come up, they get flipped on their side, and then they transported into the lab that you see here. This is called the catwalk. And so the sediment cores get put down here, and we cut them into 1.5 meter pieces, and then process each one. So these are 24-hour operations as well. Lots to do. I touched on that there's different coring systems, and we're reliant on a specific one. So what we're looking to use on these ships is a piston coring system. That's a coring system that pretty much shoots a corer into the ground, and it recovers these really nicely layered cores here that we can use for ancient DNA analysis. With a ship like this, and deep ocean operations, there's other systems on board as well, because at some stage in the sediments you might hit rock, and of course that's interesting to other disciplines as well. So you want to keep going, and then you switch to other coring systems, but they kind of whipping up the sediments a bit because they're actively drilling at the bottom, and so those ones are not so suitable. So that is why we're kind of limited to what the piston corer can do. Usually I think it's approximately 300 to 400 meters down into the sediments that can be achieved with these piston corers. So then the piston corer system is released hydraulically. So there's a pump in the middle of the ship here that basically it pushes drill fluid towards the corer to then trigger the corer. And this drill fluid is more or less seawater. So we're literally pushing seawater towards the coring system, which of course it could be a source of contamination. Normally this does not get into the core pipe itself, but we wanted to make sure, and we have applied on this expedition a technique that's very commonly used in microbiological analyses, which is the application of chemical traces. So we have this pump here that pushes the drill fluid towards the corer. We're connecting it to a different little pump, and then infuse the drill fluid with some chemical tracer. So constantly at a certain rate, we're putting that into here, and we can measure that along the core later on. So this is a non-toxic chemical tracer that we can use. And for microbiological analyses, it's been used a fair bit because they're using little bacteria from the sediments, and they're culturing them. And of course, any type of contamination there would be detrimental. So we've just adapted that basically for this purpose, and yeah, did some sampling for that. So then when it comes to the sampling, this is like a schematic here of a core that's sitting on the side, but this is what the core really looks like when it comes up, or I should say not quite, because this has undergone some cleaning already. So when it comes into the lab, we're cleaning it with bleach and ethanol, especially in the places where we're planning to cut it. And then we collected samples for the chemical traces and for the DNA analysis. So for the chemical traces, we would collect 2 samples, one at the side of the core and one at the center. And then the DNA sample collected at the center of the core as well, right next to the chemical tracer sample. So this is, if we did find any chemical traces here in the periphery, we would know that some of the seawater in the coring system might have reached the depth that we wanted to sample it. And when we find something in the center, then we know it's also penetrated into the core, which wouldn't be good. But that's the idea behind this. And we've done this kind of chemical tracer sampling all along with the DNA samples as well. So then there's a few other samples that we've collected, like samples of the drill fluid, a couple of air controls, and some swabs around the labs as well. And for this study here, I've collected about 80 samples using this approach here from the whole core sampling every 1.5 meter, because that's the kind of sectioning that's done on these 10-meter cores. And the age that they were dated at during and post voyage were up to 1 million years. So on board, we've already done some biostratigraphy analysis and magnetostratigraphy of those samples. And then after the expedition, more work has been done to refine these initial ages that we've generated on board. So just quickly on the chemical tracer analysis. So this was also done on board. Like I said, the ship is huge facility of all sorts of labs, and including a chemical lab and a chemistry lab. And so we've run the chemical tracers right on board. This was done with the help of one of the technicians on board, and so we used the gas chromatograph to do this work. The samples were analyzed in batches, like continuously when we collected them. For one of the sites, we had much more samples, and so those were divided up in batches, and the results of that were that 2 of the sites had no traces at all of the chemical tracer. And then 3 samples of one of the sites, U1538, which was the high resolution site, had 3 samples where we found that low amounts of that tracer, and where we decided to just look at these samples a bit more carefully. Okay. So then on the ship, we just froze the samples that we collected, and they were transported to the lab. All of this work was done at the University of Adelaide, Australian Centre for Ancient DNA facilities. In the process of extracting, I've introduced a number of extraction blank controls. All the samples were randomized as well, to make sure there's no systematic extraction error that could be introduced. And then we've applied a protocol that we've optimized before, specifically for marine eukaryotes. That gives us a very wide breadth of taxonomic diversity, hopefully in the end. And then the library preparation protocol is more or less the protocol by Meyer and Kircher that was published in 2010. And then the sequencing approach here was Illumina sequencing. So these were shotgun samples. And we did a NovaSeq run for these 80 samples. So like I said, what we chose here to do was shotgun sequencing, just because we wanted to get like the first broadest overview of what's in these samples to start with. And so it's very useful to do this kind of approach if we want to look at pretty much everything that's found in the DNA sample. But it's also useful because it keeps things in proportion, so we can look at the data semi-quantitatively later on. And then also important is that it's DNA fragment size independent, so we can look at fragment size distribution, frequency distribution, and look at the DNA damage later on as well. So there's another approach called target or hybridization capture or target capture, where we can look into specific organisms in more detail. And I'll talk about that in the second part of the talk. But just to get back to the authentication part here, sorry, I'm just moving the little window around sometimes in between. Not sure if you can see the mouse for that. But anyway, when we look at the fragment size distribution of an Antarctic sediment sample, we find that most of the sequences are very, very short. And if we want to use this sort of distribution for looking at whether our samples show or as an authenticity criterion, we would assume that the deeper we go into the sediment, the further we go back in time, the shorter the fragments get. And so this is kind of like a simple idea of how we could check whether what we have is ancient DNA. But it's not quite that simple. And Caitlin Salway has written up a little paper on that earlier this year. So it's really much more complicated than that. And that's why we do some DNA damage analysis, which is a bit more robust to look at authenticity. So when we have these DNA fragments, the ends of it are vulnerable, and they are prone to damage. And this is something, the substitutions are something that we can measure with bioinformatic tools. And so what we would get when we look at the computer and we measure that and we visualize it is something like this graph here, a U shape, where we would see a higher frequency of mismatches at the end of a DNA fragment. So we've applied this type of damage analysis to the Expedition 382 samples, used a program here that's called HABbaits1 that's previously been developed to look at damage in metagenomic samples. And so what we can see here, we're just looking at the eukaryote part, but the eukaryote damage increases with depth. We've done for all the different sites. We've done some correlation analysis with some of the geochemistry parameters that we measured on board. And this was kind of like quite a initial study into this. I think, you know, there's much more to explore, but what we found here was that there's some clear correlation with indicators of organic matter decomposition, so ammonia, alkalinity, phosphate. And this is all very interesting in itself. And I think, you know, much more could be done here to explore all these different little relationships, which would be really interesting. What we found is approximately 25% of this, up to 25% eukaryote damage. Again, this is something that's only been done a few times. There's not much comparison, and we don't really know yet. Is that you know where it stops? Is this kind of like a phenomenon that we can find in different areas of the ocean? I know from our Tasmanian sediments is very similar. There's like this, seems to be this plateau at about 25% eukaryote damage, but again, this is something that would be great to explore further in the future. But then as far as the community composition goes, in terms of data processing, I used adapter removal to demultiplex. There's a couple of filtering steps like deduplication, removing low complexity reads, and then the alignments were done in MALT. And the visualizations in Megan. There's a free version, community edition that can be accessed. And so that's what it looks like. Afterwards, we get this huge tree. This is just phylum level of the starter. So you can just imagine when this is at species level, it's kind of like, you know, blowing out everything on the screen. Yeah. So, you know, you can see there's lots of data here, archaea, bacteria, eukaryotes. The eukaryotes are what I've been focusing on here. And I guess when you're looking at these trees, they would vary probably a fair bit depending on which database you use. So for the alignments. So when we were thinking about which database to use for this specific data set, there were a couple of criteria. So first, we really wanted to use a curated database because we were looking at these very, very old samples and wanted to make sure that we're using curated sequences for that. And then we wanted to look at phytoplankton, zooplankton. So these kinds of organisms, they're quite well represented in the silver SSU and LSU database. So we've run the data against both of those and then did some comparisons, because some organisms are better represented in the LSU database and then others in the SSU database. There's small differences only when you start to compare them, but they are there, and both got picked up. So in the end, we decided to make the most of the data that we've got and use both of them. We just combined the database and then ran the data against this combined database. And then we're looking at the results of those on a relative abundance scale. So, and this is kind of a summary of those results. Well, what you're seeing here are the depth profiles of the different stations and the eukaryote relative abundance composition. So you can see we've got a few less samples for U1534 and 36. There's a bit of a reference map here, what is where. And we had the most samples. That was kind of just a result of the way I could do the sampling and where the sections were suitable for sampling, really. So we had much more samples here at U1538. And then the post-cruise dating showed that some of those samples were up to 1 million year old, which was really exciting, because we did get these eukaryote signals out of these samples. So this is the oldest marine eukaryote record that we have from Antarctica and from not aware of any others for those organisms. And so what we've got here also is that we found this transition to marine higher abundances in diatoms at about 14,500 years ago. So you can see this olive color here is showing you less than 10% of diatoms were part of the marine community before that time. And then suddenly there's this huge increase towards more than 50%. So this is a time that coincides with some short-term warming in Antarctica. It's quite a well-known time following a meltwater pulse where this region was undergoing quite some significant changes, and we can see this being picked up here, not just in the relative abundance of the diatoms, but also in the composition, because we found some higher influence of open ocean diatoms than at that time, looking into this in more detail. So moving away from the composition part, what we also integrated here was, rather than just looking in the taxonomic genes, also we wanted to see if there's something we can say about productivity at the time. And so this is referring back to a study that was published earlier this year by Juan Pirela Carlosich. And this was based on a Tara Oceans data. So it's a global ocean phytoplankton sampling program, and they're sampling on a yacht all around the ocean, creating really, really extensive data sets of phytoplankton. And so they've developed a database of PSBO, which is a photosynthetic gene, and it only occurs in a single copy in each phytoplankton. So we can look at abundance of past phytoplankton here. So that this is really interesting. But because it's only one copy, as opposed to multi-copy genes like the SSU, the LSU, we're also only finding very, very little of it. So this is the same plots, and you can see just here by the abundance of reads, we're reaching approximately 10 reads for the same time, it's a bit limited in that sense. But it is interesting to find the PSBO gene tied to diatoms, so it is originating from open ocean diatoms around the same time. But yeah, going back to the rarity of this, so this is where this technique, hybridization capture, becomes so important, because we can target these rare genes and amplify them before we even sequence them, and then we can look into more details. So this would be really interesting to do, for example, to this gene or other genes of interest. So yeah, while metagenomics, what we've just done here, gives us kind of a broad overview of the communities of the past. When we use the target capture, we introduce little RNA probe that we can design the way we want them, whether that is matching a taxonomic gene or a functional gene or a whole genome of one single species. We can design them so they're complementary to any type of DNA that we'd like to amplify, and then have a more detailed look into. And so this is something that we're looking to apply to these samples and other samples from Antarctica in the future. There's one bait set that we basically have ready to go. It's called HABbaits1. We've developed that previously, and it's got baits that are made based on the 18S V9 region of lots and lots of different phytoplankton and their predators, and also 3 widely distributed cyanobacteria. Then we're looking to apply the same approach for individual species or groups of species. So for example, we're really interested in the evolution of sea ice diatoms, and Prashasti Singh, who is a PhD student here, she's working on developing those. And then we've got a project that'll develop the krill baits, basically. So baits to capture Antarctic krill, which is kind of the most important zooplankton in the Antarctic region. So yeah, those are projects that are currently starting. But unfortunately, I don't have much data there to show you yet. It's all in the starting phases. But what I can show you is how we've developed a different bait set and applied it to some organisms to capture more details about organisms here along the Tasmanian east coast. So this is a study where we've been looking at harmful algal bloom species, or in short, they're called HABs. There's 3 dinoflagellates here in the Tasmanian region that are quite a nuisance in recent years. So there's Alexandrium catenella. This one is believed to have been here since a long time. But it's increasing in abundance in the last 10 years and creates these toxic blooms. Then there's Gymnodinium catenatum. This is a very highly toxic dinoflagellate. It's been thought to have been introduced in the mid-1970s here by ballast water. And then there's Noctiluca scintillans, which is a red-tide dinoflagellate. It's been introduced from the northern regions of Australia, traveled down with the strengthening East Australian current. It's even been found in the Southern Ocean in recent years, and it's a dinoflagellate that doesn't form a cyst. So it'd be really difficult to detect it using microfossils, because it just doesn't have any skeleton, whereas the other 2 are medium good cyst producers, so we can find them in the sediment record. But yeah, results from sediment records are inconclusive so far, and this is why we've tried to apply the Baits approach here to capture these 3 dinoflagellates in a sediment core from Tasmania. So I thought it probably I just describe a bit about how we developed this bait set. So I manually collected lots of different sequences that I could find of these 3 dinoflagellates online. So these included lots of different genes and gene regions. They included the LSU, the SSU, D1, D2 LSU region, COI, RBCL, ITS, and even some saxitoxin A genes. So pretty much everything we could find for these 3 dinoflagellates. And then assembled all of this into a file that was the basis for the Baits design. And then we collaborated with the company HABbaits1 in the USA, and they were basically helping us to make this into a bait set. So they're super helpful in case you're looking at doing anything like this. I really highly recommend to work together with them. So what then happens is these sequences that we want to base a bait set on, they undergo all sorts of filtering steps until they are synthesized. For example, HABbaits1, they have their own reference databases they can utilize and then blast the baits, the sequences of the baits against target taxa, non-target taxa. And then if they would capture anything or match anything that is a non-target taxon, that would be removed from the bait set. Now, so there's different things they can do to make these baits as specific to the organisms as possible. And that's a process to work through with them. So at the end, we had about 15,000 baits in this bait set. They are 80 nucleotide baits in the end. They get sent to you as a little vial and as a kit that can be applied. So as I mentioned, samples we've applied this bait set to came from Tasmania. So there were 2 sediment cores that were collected in 2018 with the investigator. So this ship has the capacity to collect multi-cores. They're about half a meter long or a meter long, but the sediment cores we got were approximately half a meter long. And so the short cores we had from inshore Maria Island. This is like a little island off Tasmania. And then a longer gravity core that was about 3 meters from offshore Maria Island. And so we had these samples frozen, applied DNA extractions with the same protocol we used for the IODP samples, then prepared shotgun sequencing libraries. That was all done exactly the same as what I've just shown you. And then a subset of those samples we applied the baits to. And yeah, at the end, we sent these samples away after we've done all the cleanups. We're also selecting for the smaller DNA sequences. So everything that's larger than 500 base pairs is removed. But to give you a bit more detail about this and the protocol, so on the left here, you can see the procedure, and this is pretty much copied from the MyBaits manual, but just to show you sort of the idea of what happens in the hybridization capture reaction. So at first, you're basically mixing all the reagents, your sequencing library, some adapter blockers, and the hybridization reagents. Then your libraries get denatured, and the adapters can hybridize. The baits get introduced, and then the hybridization reaction in itself takes place. I'm not sure what the minimum time is. I think it might be 16 hours, but I just thought here to point out we did this for 40 hours. So this is a very long protocol, just to keep in mind when you're doing this. It runs over approximately a 2-day period. And then the bait hybrids, they get bound to some magnetic beads in the next step. So this is how you basically you're kind of capturing your target sequences, they get bound to some magnetic beads, and then there's a couple of washes that get applied, and then in the end you can elute the DNA, which is then should only be the DNA that you were targeting for, and everything else gets washed away. Yes, so just here I wanted to point out in this kind of step again, there's a bit of the beads come in a certain solution to make sure they're preserved well until the time that you want to use them, and there's a bit of an extensive washing protocol before you can apply them. So again, something to consider in terms of time. So I pretty much followed the manual's instructions here, but we've done some modifications which I thought might be useful to point out. So normally what is recommended for as input template DNA is about 100 nanograms of DNA. We did not reach that with our samples. We have very low amounts of DNA in these samples, and so even though I did some additional amplifications beforehand to try and reach 100 nanograms, I never did. But I was able to apply this protocol reaching approximately 62 nanograms as well. So just in case you run into this problem. 7 microliter per library is what's required as input, and for the extraction blanks I've pooled a microliter together to make it one reaction. It is not a very cheap protocol, so that is kind of one of the reasons behind this, but certainly worth to still process some extraction blank, even though that is the case. So the recommended hybridization capture temperature that is recommended by Arbo Biosciences is about 60 degrees. This is probably something that I would do if I was going to hybridize with the plankton baits, where I'm looking to hybridize any sequence that is similar to what I'm looking for to get the broadest possible capture. But if I want to look for very, very specific sequences, and I want to make sure that really that particular sequence and that particular dinoflagellate, for example, is captured, there's things you can do to make that more specific, for example, increasing the hybridization capture temperature. So for the dinoflagellate baits, for example, we set the first 3 hours to 65 degrees to have a more specific binding, and then reduced it to 60 degrees afterwards. And that'll depend on how specific you'd like the binding to be. After the hybridization capture reaction is done, the libraries get amplified again, and then they get sent to sequencing. And what we did in this case with the dinoflagellate baits was Illumina sequencing again, our next seq run. So in terms of data processing, I didn't do much different than to anything I've already explained for the Expedition 382 samples, although because we did have this various mix of genes that we were targeting, we chose the NCBI database as a database to compare with, just to capture any potential genes that we were targeting in one go. We also did some damage analysis of this, and again we found for the offshore core some damage of those particular dinoflagellates that were up to 27%, very similar to the previous results. The inshore core is much younger. It's only about 140 years old, and so the damage patterns here were much lower, which was expected. What we've done additional here, because we're looking into 3 specific dinoflagellates, we also did some dummy runs in our bioinformatic pipeline of sequences of these dinoflagellates to get a better idea of how good the assignments are for these specific species, because we're dealing with some quite short sequences. So what we've done here is we took sequences of the 3 dinoflagellates from NCBI and their genotypes, and then used the whole sequence, but also the sequence chopped up in 56 base pair fragments, and then ran everything through our bioinformatic pipeline to be able to assess whether, how good the assignments are. Can we make them on species level for these 3 species, or do we have to be a bit more conservative? And so for Alexandrium, we found that we can only make species assignments on genus level. For Gymnodinium, we are confident we can make them on species level, but we had to do some, even on top of this, we had to do some extensive additional verification of these sequences and map them back to reference sequences to see where exactly in the genome they match the reference sequence. So that there was quite some intensive work that went into this. But yeah, we're fairly confident here we can do it on species level, and Noctiluca scintillans has a very unique genome, and that was we're quite confident there that can be done on species level as well. So it's quite different to the others. In terms of results, I just briefly say something about the shotgun data that we generated from this here. We had a total of approximately 800,000 sequences generated from this. But what I want to point out is only about 500 of those were for dinoflagellates. So that's approximately 0.06% of all the data, so including eukaryotes, bacteria, and archaea, and 0.35 of all eukaryotes. So when we then go to the hybridization capture results, we can see these ratios, they much improve. So we have had approximately the same output for sequences. We have 32,000 sequences that are now assigned to dinoflagellates, 5% of all eukaryotes. So we do get a significant increase here in the efficiency of capturing the species that we're after. Then in terms of ecological interpretation of the data that we had here for Alexandrium and Gymnodinium, we did find sequences of them all the way through in both cores. Alexandrium, though, especially increased in relative abundance in the inshore core. So it does look like recent promotion of growth of this diatom in the region, probably due to the changing conditions that we're seeing here on the Australian east coast and Tasmanian coast. Gymnodinium catenatum, like I said, we're still verifying this particular species, and it's whether we're looking at a group of microreticulate species that include Gymnodinium catenatum. But yeah, at this point, we're fairly confident it is this species, and it does occur all the way through since about 9,000 years. So this means that its recent growth is probably also likely due to changing environmental conditions, and it's been around. It's just not been detected before using other analysis. For Noctiluca scintillans, we also found some sequences of this fragile dinoflagellate, especially in the last couple of years, since about 2010, and that matches observations of this dinoflagellate being introduced. At the very, very bottom here, we also found 1 or 2 sequences, which needs a bit further investigation, just because it is possible this is a sample that's collected at the very, very end of the core. And in this case, we don't have chemical tracing. There is a possibility there's some seawater contamination there. The verification steps also showed that there's some potential misassignments with other dinoflagellates. So again, this needs a bit an additional verification here, but yeah, that was all I had prepared for today. So just a couple of summary points here of what I'm going through, what I've been going through in this talk. And yeah, if you have any questions, please ask.
Hi, Linda. Vilma here.
I thought I couldn't hear anything. I panicked.
Thank you for that presentation. That was great.
Yes.
I wanted to ask you about the analysis that you did with the 56 base pair fragments. I couldn't really understand. Sorry, I lost the audio there for a bit.
Yeah.
Could you explain that again very briefly?
Yeah, of course. Let me go back to that slide too. So when we ran, we had the shotgun data already for the set, and calculated the average size of the fragments that we had, which was 56 base pairs. So from there, decided we chop the fragments up into 56 base pair sizes. So for those 3 dinoflagellates that we were targeting, you basically see all the representatives here and the sequences that we used. So I used just downloaded different sequences of different genes of those individual taxa. The whole sequence would have been, in this case, 575 base pairs long, 1,755, and so on and so on and so on. So we'll include this into the analysis, but also a chopped up version of this, where we have fragment 1 of this sequence, fragment 2 of that sequence, fragment 3, and all of them were 56 base pairs long. And so then we basically used this to run the exact same analysis and run it against NCBI, and then check the output of that to see how reliable the assignments are. And you could see, for example, in the Alexandrium tree, it would usually stop assign, or it would be assigned at Alexandrium genus level and no further. And then there's maybe the 1 or 2 odd sequences that got assigned to different dinoflagellates. Or with Gymnodinium, it could get assigned on species level, but there were a few more additional dinoflagellates that it got assigned to, which shouldn't be the case, because you literally just input a Gymnodinium sequence. And so based on that, we kind of like made the decision in terms of interpretation cutoff for these particular results. We interpret Alexandrium at genus level, Gymnodinium species level, in the context of the microreticulate group. That's a group of Gymnodinium species together with 4 others, and that are really similar to each other genetically. And then Noctiluca scintillans, that is easily basically back assigned to the same reference sequence that you chose from the beginning. Did that explain? Did that answer the question?
Yeah. Does the same problem is presented like with modern data? It's just those species are really, really difficult to classify? Okay.
Yeah.
Okay.
Yeah. And because they such disruptive species and potentially very toxic, it was quite important in this case to go into these details and check those sequences and see how long which one's been present in this area.
Yeah. Okay. Yeah, there is a similar issue with like eDNA and those pathogenic species. They're really difficult to assign to the strain or species, even species level.
Yeah.
So it's very important that you're very like careful of how
Yeah.
you're gonna differentiate it. It's great that you did that here.
Yeah, and especially with Alexandrium, you know, there's for example, there's certain strains even that are toxic and others that are not.
Yeah.
And you know, it would be quite important to find out if you can go to that level of discrimination, or you can't really. You know, you have to differentiate at just cut off at a higher level. So
yeah. Okay, thank you.
Cool. Thanks for a fascinating talk, Linda. Apologies, I was on mute before, but I do have a question. The use of chemical tracer versus DNA-based tracer, what was the decision there? Did you consider using DNA tracer as well?
Well, I've heard about them. I haven't really considered them at the time that we were on the expedition. I do not know much about them, to be honest. With the chemical traces, so there's just two approaches that have been used frequently before on these kinds of expeditions, and one is microfluorescence particles. You can see them in microscopy samples, but they are quite big, so they probably wouldn't, you know, mimic DNA at all in terms of how they might travel or how they might enter the sediment core. Chemical traces have been used a lot in this context, so that was something that we had available and we could implement relatively easily. I mean, it was still a bit of additional logistics, of course, but something that could be done and with what we had available and so on. Yeah, DNA traces, I'm not too familiar with. Have they been used for in other contexts?
It's something we often use when we're just collecting cores on terrestrial sites, and it's just a matter of spraying down the core or an equipment with the solution of a known DNA fragment that you can then try and detect through PCR later on. But same sort of idea as a chemical tracer, just sort of kind of a different approach. But no, it was really good to see that you didn't detect it in a lot of your samples as well. So I might just quickly throw in one more question. I was interested in the fact you picked up chordates. Did you look at those in more detail to see what they were? Were they fishes or penguins, whales?
Oh, you mean higher organisms?
Yeah.
I haven't yet, no, in detail, but yeah, I mean, there is certain fish DNA that I know, and in Tasmania we did actually pick up a orca sequence once, which is very exciting. But yeah, it would need some looking into. So yeah, I mean, it's just really small amounts, but it would be so interesting to look into. Yeah.
And maybe also if there's terrestrial signals in there as well,
Right.
plants, maybe, you know, I guess did anyone work on pollen from the cores or?
No, not really. Dinoflagellate cysts, diatoms, yeah, those kinds of, you know, typical microfossil biostrat organisms, but not pollen so far. Yeah. Yeah.
Do we have any other questions? No, okay. Well, I think thanks again for an awesome talk. We'll put the link up online shortly, and I'm sure that a lot of people will watch it, and you might get a lot of emails with additional questions as well. So it really was a fascinating talk. So thanks very much for that.
Yeah, thanks so much.
Okay, bye everyone.
Bye.